Hi guys, you're watching my YouTube channel BioHub and I'm your facilitator Dr. Seema Sharma. Aim of this activity is to show that light is necessary for photosynthesis. To carry out this activity in our science lab, we need certain glassware and chemicals. So let's know about the requirements. We need a potted green plant, iodine solution, a pair of scissors, black paper, stencil and paper clips. In addition to these things, there are certain other requirements: beaker containing water, test tube with test tube holder, Bunsen burner, wire gauge, tripod stand, ethanol, forceps, petri dish and lighter. Now coming to the steps. First step is to take a potted plant and de-starch its leaves by keeping it in dark place for 2 days. You know viewers, during photosynthesis glucose is formed. This glucose, it is converted into starch in the leaves and then it is transported to each and every part of the plant. When we are keeping our plant in some dark area, then the chances of photosynthesis will be very low. During that time, the stored starch, which was meant to transport to different parts, it will be reconverted into glucose and it will be utilized by the plant for its basic requirements. So after 2 days, these leaves, they will not have any starch in them. So this is de-starching. Second step is take a black paper and cut out any letter. In this activity, we have cut out a letter L using a stencil. Third, cover one leaf of this plant with this black paper without plucking it. Fourth step is to shift this experimental setup in bright sunlight for 5 to 6 hours. And in fifth step, we have to detach this leaf with the black paper and test it for the presence of starch. Step number 6. I have divided step number 6 into 5 sub-steps. In first sub-step, we have to keep the leaf in boiling water for 5 minutes. After boiling, this leaf will become soft. We have to put this softened leaf gently in test tube containing ethanol. In third step, we have to keep our this tube in water bath for 7 to 8 minutes. And this step should be performed very carefully because ethanol can catch fire very easily. After this, in fourth step, we have to take out this leaf and we have to rinse it with water. Thereafter, we have to spread it very gently on a clean petri dish and pour iodine solution over it. And then we have to see, is there any change in color? This iodine is a universal indicator of starch. In whatever material starch is present, if we are pouring iodine on that, immediately it will turn blue-black. Observation. This is the leaf before starch test and this is the result of starch test. So observation will be that the non-exposed part of the leaf will not show any change in color with iodine solution, whereas part of the leaf that was exposed to sunlight, the tip part, the basal part and the cutout of letter L, they were exposed to sunlight. So here iodine has reacted with the starch and it has given blue-black color to the leaf. So on the basis of these observations, we can conclude that no reaction with iodine in the covered part of the leaf shows absence of starch. On the other hand, change in the uncovered or exposed part of the leaf to blue-black color shows presence of starch. This clearly shows that photosynthesis took place in the uncovered or exposed parts of the leaf where light could fall on this leaf. With this activity, we can prove that light is necessary for photosynthesis. Precautions first. Before starting the experiment, the leaf must be de-starched. The leaf must be covered with black paper properly to prevent the entry of light. Boiling of the leaf in ethanol should be done in the water bath and that too very carefully because ethanol is inflammable. All the glassware should be properly cleaned and wiped before and after the experiment. Last but very important precaution is wear a lab coat while performing these experiments in your science lab. Now I'm sure you are ready to go to the lab. So keep experimenting and continue learning. Good luck.